Thank you
Okay,
very much, Ned, for agreeing to give a Build a Cell seminar, and you can get started.
great. Well, thanks everyone for showing up to this, and to those who are eventually listening to the recording. Thanks for tuning in. I'm Ned Wingreen here at Princeton University. Quick bio. I started out doing condensed matter theory. I had no idea I was going to end up doing anything biological. I might have actually taken a course in biology somewhere along the way if I had known that. But somewhere in my career, I started fooling around with some simple models for proteins. I'm a theorist, so I like lattice models, you know, and there were only two types of amino acids in those models, too much to remember as far as I was concerned. And somehow that started me down a slippery slope. And I've ended up in a molecular biology department here. And I just fell in love with biology. I think it's so rich, and biological systems are constantly using physics in creative ways. And so my group has a few areas that we work on. One of which has been microbiology, which for many years was our bread and butter, how bacteria find each other and find food and talk to each other. But in recent years, we've also become interested in this area of intracellular phase separation, which I think is another great area where physics and biology are, really close, coming hand in hand. And so that's what I decided to talk to you about today, particularly the problem of condensate size control. How do cells, or how could we, in the context of Build a Cell, learn to control the size of these intracellular condensates? And of course, I'm going to tell you what these condensates are, and I realize it's not going to be familiar to everyone, but it should be, because they're inside your cells right now doing important things. So let me begin with an outline briefly to what are these biomolecular condensates in our cells. And then I'm somewhat ambitiously going to try to cover two topics in this area. Part one will be active control of condensate size, with a particularly paradigmatic example of something called the pyrenoid that appears in algae as part of their photosynthetic process. And then in part two, something more speculative, whether we can actually design self-organizing enzyme condensates that would function better than dispersed enzymes. And then, given a few minutes, I will conclude at the end. So let's dive in. What are these biomolecular condensates? And so first off, I should say that they are ubiquitous. This is a bit of a rogues gallery of different condensates. Every month or so, there's a new paper discovering that some particular pathway in eukaryotic cells, or even in bacterial cells, is involving a condensate that we didn't know about before. Up in the upper left, you'll see those green dots. That's a C. elegans cell about to undergo its first division, and material has been partitioned into one daughter cell as opposed to the other by a process of phase separation. So there's many different components to those yellow or green dots, including a fluorescently tagged protein that has been engineered in there. But in that case, this condensate is being used to get material into one lineage as opposed to the other. Just below that on the left, that's from a human cell line. And the cell at the top has been starved for purines, the A's and G's in our DNA and RNA. And in order to make new purines, little enzyme clusters have formed in the vicinity of mitochondria, where the substrates for that production are localized. And those are then processing those molecules inside of little clusters of enzymes and pumping out new purines. And if we feed that same cell purines from the outside, it no longer needs to produce its own, and those little enzymatic factories disassemble, and you see it's dispersed. And so there are other examples where condensates are playing a crucial role. If you move to the one more to the right on the top, that's involved in signaling. Lots of cases where condensates are playing a role in signaling, including in T cells, which are running around your body right now, making sure that your cells are healthy. And certainly in in gene regulation, both repressing and activating genes. There's quite a bit going on in terms of condensate. So if you look at the upper right, you'll see that rather complicated looking object with red and green. That's a nucleolus that's inside the nuclei of our cells. That's where ribosomes are being manufactured in order to the ribosomes translate mRNAs into proteins. We need a lot of those. And down to the right, you'll see what happens when two of these nucleoli get close to each other. And so even though each of these has lots of components, RNAs, proteins, many different types, if you look at what happens when they touch, it looks relatively straightforward from the physics point of view. There's two spherical droplets. They're spheres because of surface tension. That's the lowest free energy state. When you bring them together, they realize that the lower free energy state would be to merge into one bigger droplet. And that's exactly what they do. So even though there's a lot of biological complexity here, there's some underlying physical simplicity, that these do behave like phase-separated objects, more like simple things we might have dealt with, like oil droplets in water. And so the hope is that we can understand what's going on using physical principles. So yeah, so that's background by way of motivation for this field. And I should say this is far from the only set of condensates in our cells. As I said, there's new ones being discovered every day, and there's order 50 or 60 that are known now, with more coming. So watch this diagram. All right. So the presence of all these condensates raises the question of, how do cells organize their condensates in space and time? And so there are a lot of things the cell might want to control the number of condensates of a particular type, say the number of nucleoli or the number of these purinosomes that are making purines, might want to control their location. In this case, showing those condensates for some reason want to be near the cell nucleus. The purinosomes want it to be near mitochondria. So this could be very important for their function and also their size. We did some work some time ago on enzyme clusters, and it turns out there's a preferred size for those. If they're too big, then they get starved for substrates. If they're too small, then the intermediates escape. So again, all of these things could be very important for the function of condensates. And so cells, presumably over the course of evolution, have learned how to control these properties of the condensates, their number, their location, and their size. So what I'm going to talk to you about today is this example of the algal pyrenoid, which we've been studying theoretically, but very much motivated by the work and my colleague Martin Jonica's group here at Princeton, who is the world leader in studies of this particular organelle because of its function. And I'll tell you a little bit about that. So here is, for example, the straw man that I'm going to set up and knock down. This is a poor plant cell whose chloroplast is filled with this enzyme rubisco that converts CO2 eventually into organic carbon compounds and eventually into sugars. And this poor plant cell hasn't figured out how to be very efficient about this. And so it needs lots of rubisco spread through its chloroplast. And in fact, this enzyme rubisco is, I believe, the most abundant enzyme on Earth. So plants are producing tons of this. It's a real big cost to them. But here's the clever algae that has figured out that if it can put all of its rubisco together in a little cluster and then pump CO2 into the vicinity of that cluster, it can get more bang for its buck from each of these rubiscos, and therefore needs to make less of them and invest less resources into the rubisco and grow faster. And so Martin has the goal of engineering these pyrenoids into crop plants like rice and soybeans and various forms of wheat in order to improve yields. And he's organized an international collaboration that's making real progress on this. But along the way, one runs into lots of biophysical questions about how this actually works. And so we've been collaborating with Martin to understand those, and we think that's going to help eventually with his goal of engineering these into crops. So the idea is with this higher level of CO2, the rubiscos are then saturated, they're working at full efficiency, and therefore they'll be more efficient at fixing CO2 and turning it into sugar, which of course animals like us rely on. We need the plants and the algae to do the carbon fixation. So this pyrenoid where this happens is really a model for phase separation. And so different algae do it in slightly different ways. We're studying the one, a model organism called Chlamydomonas reinhardtii, or clammy for short. And clammy does the following. It organizes this pyrenoid, which in algae mediates about 30% of total global CO2 fixation. So plants are doing a lot, but algae also. There's a singular condensate in clammy. So as I've shown it, there's one cluster, and that's really correct. It's not like there's a whole bunch of them. There's one in its particular location where the CO2 is going to be delivered. So it has to be in the right place. It has the advantage that thanks to Martin and others, the drivers of this phase separation are well understood. There's this enzyme rubisco, which is sort of these spherical rigid objects, but those are held together by an intrinsically disordered protein called EPIC1 that has binding sites, multiple binding sites for the rubisco. The rubisco by symmetry has 8 binding sites for the EPIC1, and so they phase separate when you put them together. And so that's what this looks like, of course, very schematically, that each rubisco could be interacting and bound to multiple different EPIC1s. Those are the little flexible brown squiggles. And each EPIC1 could in principle interact with multiple rubiscos. And so because of this multivalent interaction, when you put them together, they phase separate. And this is a theme in this field of condensates, that they're often driven or held together by these weak multivalent interactions. So in this sense, the pyrenoid is very characteristic of other biomolecular condensates. So there's the rubisco, there's the linker protein EPIC1, and they're doing their thing inside the cell. This is an admittedly computer schematic of what's going on. Those are the right atomic positions for the rubisco. The binding sites are the right atomic positions for the EPIC1s, but we don't know what those linkers are doing exactly. So those have been sketched in. But you see, that's the idea, that you'd have these flexible EPIC1s bridging interactions between the rubiscos, bringing them together. But of course, that doesn't tell you how they're going to get to the right place at the right time. And there's more to come, more surprises about this particular condensate. So as I've said, there's these nice properties of it, but something else that I didn't mention is that this condensate, the pyrenoid, is actively regulated. And what I mean by that is that every time that cell divides, it actually dissolves the condensate into the chloroplast, spreading out the rubisco and the epic. And then when the cell divides, it reforms in the daughter cells. So there's some very regular process of formation and dissolution. And so let me show you a little movie. In this case, the elements of the pyrenoid are blue. The rest of the chloroplast is glowing green. And if you watch, you'll see that as the cell is about to divide, that condensate is dissolving. But as the cell goes through divisions, it's actually going to go through 2 divisions. That's just the nature of clammy. It will reform. But notice that as it divides, there are other little pyrenoids or little condensates are forming at the same time as the first one is dissolving, which we'll come back to. It seems a little bit counterintuitive. If the cell has organized things to dissolve that first pyrenoid, how come they're actually able to reform in other places in the cell at the same time? At the end of the day, and when this stops, you end up with 4 cells, each of which has 1 pyrenoid, which is what the cell wants to accomplish. So something interesting going on here in terms of the regulation of the number of these pyrenoids, 1 per cell. All right, we stop that and ask, how does the cell regulate these dynamic phase transitions? So the answer is that we have a very good idea now about how that works. The Jonicus lab has identified a pyrenoid-associated kinase that they called Ki1. A kinase is an enzyme that adds phosphate groups to its substrates. And in this case, its substrate is this linker protein EPIC1. And so what's happening is that Ki1 is adding phosphate groups to EPIC1, and that's having an effect on how well EPIC1 sticks to the rubisco. And so with Ki1, that's what dissolves the pyrenoid. EPIC1 gets phosphorylated. Without Ki1, the pyrenoids don't dissolve during cell division. So that's a pretty big clue to what key 1 might be doing. Here, the cells are actually going to go through cell division, but instead of the pyrenoid dissolving, all those pyrenoids are actually maintained through cell division. So there's the dissolution doesn't happen. We know a lot more about this, but I know the time is very limited, so I'm going to only touch on some sort of key points about key 1. So key 1 is necessary for these phase transitions during the cell division. So if you look at how much of the rubisco is in condensates, in the blue curve, every time the cell goes through divisions, most of the rubisco leaves the condensate. That's those big dips. In the cells lacking key 1, there's really no dissolution. The key 1 is almost all in the condensates all the time. This is just quantifying that for your reference. It's a big effect. So we also find that EPIC1 is highly phosphorylated at stages during the cell cycle. And remember, the phosphorylated form of EPIC1 is the form that's not sticky. So it's not able to hold the rubisco condensate together. So that can be measured by phosphag gels. And one sees this is hours during the cell cycle. And I should say that clammy is very much hooked up to the 24-hour circadian cycle. It's a photosynthetic algae. It really needs to know when the sun is coming up and when it's going down in order to be efficient. And that upper band there is showing you the phosphorylated EPIC1, which is kind of peaked just when the cell is going to go through division, just at the end of the day. And if you don't have this enzyme key 1, then you don't have the phosphorylated EPIC1. So it's sort of hanging together. Key 1 is what's making EPIC1 phosphorylated. At the end of the day, that's causing the pyrenoid to fall apart because the EPIC1 is no longer sticky for rubisco. So,
hey Ned,
yeah.
there's a question in chat. The question is,
go for it.
Do we know what physicochemical factors affect the phase transition? Is it pH, some small molecule dependent?
That is a fair question. There are some effects of pH, not super strong because the experiments have been done at different values. But in vivo, the real regulation is, as we understand it, through phosphorylation of EPIC1 and then dephosphorylation by phosphatases. So, all of these things would, of course, be sensitive to other conditions like pH, salt concentration. But within the cell, the active regulation is based on this enzymatic activity. But that's a fair question. Thank you. So, but if we're right about this, then we should be able to reproduce this behavior in vitro, and that's been done in the Janicus lab, so that phosphorylation of EPIC1 indeed inhibits phase separation. So if you mix EPIC1 that's unphosphorylated and rubisco together, they will spontaneously phase separate in a test tube. But if you phosphorylate the EPIC1, then in the lower panel, there's nothing to see there. It's because there's no clusters have formed. And so the schematic on the right shows you that the phosphorylated EPIC1 is just not sticky to rubisco. Okay, so key 1 levels indeed vary through the cell cycle, peaking in particular where you see the light to dark transition, though there's some other peaks as well, which I'm happy to talk about later if people are interested. So we're hanging things together, but of course this is all words at the moment about how this works in a quantitative level. And so you may ask, well, what am I talking about this? I'm a theorist, and here comes some theory. And the idea is that this spatially delocalized kinase, this key 1, which is everywhere and binding to EPIC1 everywhere, can actually control condensate size. And this comes back to this point that we mentioned earlier, that even as the pyrenoid is dissolving right prior to phase separation, other small pyrenoids or condensates seem to be forming. And so now I'm going to try to explain our understanding of what's happening there. And basically, it's this promised size control mechanism. And so the idea is that this is active, that there's constantly this kinase key 1, which is phosphorylating EPIC, making it not sticky, and it leaves the condensate, bringing some rubisco with it. But at the same time, there are phosphatases around that are removing the phosphates from EPIC 1, making them sticky again. And so those sticky EPIC 1s and rubiscos are coming back in. So you have these fluxes. You have an outward flux shown by these red arrows, and at the same time, an inward flux of sticky versions of EPIC 1 and rubisco coming in. And so you can imagine that there's a total flux, and the condensates will only keep the same size if those are in balance. So you can imagine that if those total flux is 0, the condensates are sitting there the same size, but you could adjust those fluxes by changing the rates of phosphorylation or dephosphorylation. So just schematically imagine that you increase the rate of phosphorylation by increasing the amount of Ki1, then you will have a higher rate of EPIC 1s becoming phosphorylated and a bigger outflux of these molecules, which will then make these condensates shrink. But then if you come in and change that, reducing that again, then the condensates can grow again. And in fact, you can think about it, well, there's a total volume of this condensate that goes like its radius cubed. So if this phosphorylation is happening everywhere, and we see Ki1 localized everywhere, including inside the condensate, you'd have an outflux that goes like radius cubed. But then you'd have an influx that if it's diffusion limited goes only like radius, or if it's reaction limited goes like radius squared. An r cubed and an r squared coming into balance would happen only at a particular value of r. So there's a preferred size of these condensates. And what we imagine happening during cell division is that initially that preferred size is big, containing all of the rubisco and EPIC 1. But as you turn up the activity of Ki1, the preferred size becomes smaller. So even as that big condensate is shrinking, new small condensates are forming, aiming for the preferred size. So that's what we understand happening as a size control mechanism. And I should emphasize again that it's an active mechanism. There has to be energy being used to both phosphorylate and then dephosphorylate this EPIC 1. Those are, again, kind of a schematic. We can do better than that. We can write down simple models. These are admittedly very simple models, but that's the idea, to show that this doesn't require a lot of fine tuning to make this happen. We can have an equation for the unphosphorylated, that's the sticky form of EPIC 1, a partial differential equation in space, a corresponding equation for the phosphorylated, that's the sticky form of EPIC 1. The first term in those is just the divergence of a flux, telling you that the rate of change locally will depend on the divergence of the flux, where that flux will be determined by the free energy of the system, giving rise to chemical potential gradients for the sticky form and the non-sticky form. The sticky form, of course, liking to find other sticky molecules, so we're going up gradients of other sticky molecules. That plus diffusion gives rise to these fluxes. But then the whole process is active because there's switching. So we assume some rate of switching caused by the kinase that switches from the sticky state to the non-sticky state, and then phosphatases that switch back from the non-sticky state to the sticky. And again, to be really, really simple, we assume that these are just constants everywhere in the cell. There's just a certain rate at which a sticky EPIC 1 becomes non-sticky, and another rate at which non-sticky ones become sticky. And that is actually all you need to generate a size control mechanism for condensates. That's these uniform switching rates. So just to show you then that we did some simulations, and just to be cute, we did it in a geometry that looks a little bit like the algae. The big empty place would be with the rest of the cell. The dark part is the chloroplast. And so we start with a condensate in one location, and then we change the rate. So we make the rate of going from sticky to non-sticky higher. That's like turning on this enzyme Ki1. And you'll see that while that disappears, other condensates form. That went rather quickly. So we basically turn up Ki1 and then we turn it down. I will show it again. The first condensate is going to dissolve while others are forming. They also organize themselves in space. That's another aspect to this. They try to avoid each other based on these simple equations. And so that's, for our purposes at the moment, just a kind of proof of principle that these very, very simple models of where you just have some switching between a sticky and a non-sticky state can give rise to size control. And we're not the only group that has talked about this, but I should say this is quite the simplest version where these sticking processes, these transitions are happening everywhere. David Zwicker and colleagues have done a lot of interesting work on similar models. Okay, and so that's supposed to remind you of what's happening in the actual pyrenoid, where you get the first condensate is dissolving while other condensates are still forming, essentially picking out preferred sizes. All right, from a theoretical point of view, you can end up with different regimes depending on the rates of switching and how fast the proteins dissolve. When the switching rate, when things are basically non-sticky, they don't aggregate, then you can get aggregation. Arrested coarsening would mean that there's a preferred size, or at extreme limits, you can find that there is no size control because everything would decide, just like oil and water, that it would continue coarsening, eventually getting to one big condensate no matter what. All right, so that was part one. I realize part two may have to be a little bit abbreviated, but just to summarize, this algal pyrenoid is a model condensate because we know a lot about it. We can reconstitute it in the lab, and it's a model for this size control mechanism. The active player is key one, and of course there are phosphatases that remove those phosphates to mediate dissolution and reformation of the pyrenoid. Key one is necessary, and we can understand this with a very simple mathematical model. I don't want to claim that the model captures everything about this real process, just that the underlying principle is very clear from this simple model. And so I like to think that maybe there are many other examples, and I think there are, of condensate size control via these active regulation of fluxes. And I understand, of course, for this audience interested in reconstituting and engineering things in the Build-A-Cell consortium. And so I think there's some guidance here for if you want to actively control condensates sizes as well as their number and location. So
hey Ned,
I'm
another
happy
question.
to answer questions, yeah, at this point, but also then to then see how it goes, and then we'll decide how we're going to deal with part two.
So question, I think that's for part one. Can we express the proteins that make pyrenoid all in vitro and combine it with in vitro Rubisco? We know we can express that, and study the dissolution and assembly concurrent with translation.
You know, I think the answer is yes. For the moment, the Jonicus lab does express and get EPIC1 from E. coli, so it can be expressed in another exogenously in a different organism. It turns out that's a convenient way of getting large amounts of EPIC1. So these can definitely be purified, being produced inside of cells. I'm not aware of doing this in vitro translation of these particular proteins. I don't know if there are specific challenges. I mean, Rubisco is pretty large. It's made of 2 different subunits, 8 copies of a large subunit and 8 copies of a small subunit. So I think there are probably some challenges with that. But I'm sorry, I'm not the expert who could tell you whether that's, you know, how feasible that is. Certainly purifying them from other cells and then playing around with them in vitro, adding the kinase key 1 to phosphorylate, adding phosphatases to dephosphorylate, and looking at how phase separation is affected there. That's actually been done and is going on in the lab. So that part is done.
I pasted a link in chat for everyone that answers partially the question. There's a paper where they expressed Rubisco. I think that's what Patrick meant with his question.
They express it in, okay.
Cell-free.
Oh, that's cool. Yeah, great.
Yeah.
So then the answer is probably yes, because I don't see any particular reason. EPIC1 is a much smaller protein. So if you can express intrinsically disordered proteins in vitro, then it seems like it should work. I'm happy to answer more questions about this at the end. I'm going to push on and talk about part 2 because I think that's also interesting and fun and more speculative, which I feel is good for this audience. We want to speculate about things, maybe things that we can do that even cells haven't discovered. So let me try this. So the part 2, I'm asking the question, can we design passively self-organizing enzyme condensates that organize to the right size? So not just to get, some kind of size control, but the right size. And so this is an image from a paper we wrote a long time ago about enzyme clusters. And the idea is that you might want to have enzymes clustered together to speed up the processing of intermediates. That's the big idea, but that there's a right size to do this. And so let me start by motivation that many metabolic pathways are multi-step. Schematically, you have a substrate, enzyme 1 turns it into an intermediate, enzyme 2 turns it into something else, and eventually down the way you get the product that you're actually interested in, amino acids, nucleotides, what have you. So that seems fine, but then considerations are that sometimes these intermediates can be problematic. So maybe in some cases they're toxic, sometimes they're unstable, can be degraded. They might be slow to accumulate. You really need to get a lot of them in order to get to the level where the production of the next step is fast enough. And they can undergo unwanted reactions, signaling, and of course, they always contribute to osmotic stress. There's a certain osmotic budget that cells have. And so if you fill the cell with intermediates, then the cell pressure is too high and the cell's going to bust. So how does nature deal with intermediates? So one way is to actually build a channel between enzymes, and that there are a few examples of this where an enzymatic process happens in one site, and then the intermediate never gets to leave before it somehow diffuses along some channel to find the next site, which is cool. But there's only, I would say, a handful of examples like that in nature where the intermediate is truly trapped in channel. You could imagine, oh, maybe if I just bring the two sites close together, that'll be enough, and the intermediate will diffuse between them. And that's true, but those sites have to be very, very close. Basically, the distance between those active sites has to be no larger than the active site itself, which is sort of on the scale of a nanometer. So it's not that this is not going to be easy to accomplish. Just for example, tethering two proteins together. If they're more than a nanometer apart in the active sites, then this intermediate is more likely to escape than to find the next enzyme. And so what else can be done? And the answer seems to be that you can get channeling without channels. And here's the example, again, is the pyrinosome, where you find these clusters of many copies of these enzymes clustered together. And the idea is that a substrate diffuses into this cluster, it gets processed by the first enzyme and released, and then it diffuses around, but it's diffusing around in the vicinity of a very high density of the next enzyme in the pathway. And so before it escapes, it's very likely to be processed by one of these second enzymes, not necessarily the one that was closest to the first enzyme that actually processed it. And you can see that whether this works will depend on things like, well, what's the density of the enzymes there? How fast do they process intermediates? And how big is this cluster? If the cluster is very small, you can imagine that intermediate will escape before it gets processed. If it's very large, then the initial substrate that's coming in will basically never reach the enzymes in the middle, and therefore those are being wasted. So you can imagine that there's a good size for this, if this is going to happen right. There are a number of examples of this kind of multi-enzyme clusters in cells, the pyrinosome, something called the glucosome. One can argue, and I don't want to argue too strongly, but that the transcriptional regulation by condensates has something similar along these lines. The idea, though, is that this clustering of enzymes in a pathway can increase reaction fluxes, accelerating the pathway, minimize and localize unwanted toxic intermediates, and reduce transient timescales. So there's a lot of things that could be good about doing this. And therefore, we'd like to understand how what we call these metabolons might self-organize and function, and that could be key to creating efficient new biosynthetic pathways as you're building cells. So, so the question I have is not just are these useful, but can we get them to self-organize? Because that's really an interesting challenge. Because you think of the phase separation by itself just tending to continually coarsen, and clusters would get bigger and bigger. We saw in the case of the pyrenoid that you can have a size control, but nothing there told you exactly what was the right size. Eventually, once the pyrenoid formed, the size was as big as possible. But what if these sizes really something intermediate? Could we get the system to organize itself into those sizes? And so here, in what I hope will be like the next 9 minutes, I will walk you through what we're thinking about. So here's our very, very simple enzymatic pathway. At this point, it's only one step. There's a substrate, there's an enzyme, and a product. Okay, so how are we going to get these enzymes to self-organize? Well, we're theorists. This is how we're going to engineer those enzymes. We're going to attach a squiggly tail to them. And the squiggly tail is some sticky, say, intrinsically disordered protein that will tend to make those enzymes stick together. Well, okay, that's fine. Those might tend to phase separate with that tail. But what we want them is to phase separate in a facultative way when they're needed to phase separate. And so how we're going to do that? We're going to do that by putting a divot in that enzyme. That little divot there is going to be an allosteric regulator of the enzyme. And so now follow me. The idea is that when the substrate is present, that's this little red diamond-shaped thing, it is going to stick in that allosteric site, and that sticking of the substrate in that allosteric site is going to release the tail. So if you go to the upper in this little sketch of these 4 states, what happens is when the substrate is present, it binds to the allosteric site, it releases the tail, and that enzyme becomes sticky. And so it will tend to phase separate and form clusters. Okay, so but what happens if the substrate is not present? So there's no substrate around, or these enzymes are not getting substrate. Then that site is going to be empty, and that's going to cause the tail to retract. And the tail will then retract, and no longer will that enzyme be sticky. So I am not, sorry, a protein engineer. That's my level of understanding of how this is going to work. Just schematic, but I'd love to chat with people about how to actually engineer something that will do this. But let's just imagine that this is doable. So now we have a situation where these enzymes only become sticky when their substrate is around. And that's kind of good because, for example, if I have a source of substrate, then the enzymes nearby will allosterically be regulated to become sticky, and they'll cluster around that source. And then they'll do their thing because now their substrate is present, not just in the allosteric site, but in the catalytic site. Product is being made. But that cluster should not get too big, because if it gets so big that the enzymes in the outside are not getting access to the substrate, it's all being processed, then they're not going to be allosterically regulated to be sticky, and they will just leave and maybe go to some other source of the substrate. So it looks like we might be able to get some kind of self-regulating size for these enzyme clusters. So you can imagine doing this with a 2-step pathway. In principle, the second enzyme could also be allosterically regulated, but we want to do something simpler for this purpose. We just want to make sure that wherever you have enzyme 1 clustering, you have the right number of enzyme 2. So we're going to just attach them with a short linker. So we're going to imagine that they naturally have to go together because they're engineered as one giant polypeptide. Okay, and so the idea is similarly, then you could get a 2-step pathway where the cluster is again the right size, because the first enzyme in the pathway will only cluster if the substrate is available. It will release its intermediate in the vicinity where there's already a high concentration of the second enzyme, because the first enzyme and the second enzyme are connected to each other. Okay, so time being limited, I'm going to skip very quickly through the theoretical model for that, but it's got the usual things that you would expect. It's got the enzyme densities, and the enzymes can either be sticky or non-sticky based on this allosteric regulation. So we have a free energy that's telling you that how sticky enzymes like to phase separate. This is a Flory-Huggins, very simple version of the free energies associated with entropy of mixing, intermolecular interactions, and an interfacial energy, so that interfaces aren't arbitrarily sharp. And then we model that with a Cahn-Hilliard type of approach, where essentially proteins just go down their chemical potential gradients with some fluxes, as well as diffusing. In addition to that, we do allow the switching, of course, between the sticky and non-sticky states, which is allosterically regulated by the concentration of substrate. So we need some equations for the substrate, which is itself able to diffuse. Maybe there's a source of substrate. We're interested in cases where the substrate might decay. And of course, the whole point of this is that that substrate can be catalyzed, turned into an intermediate by the local concentration of the first enzyme. And if we want to consider an intermediate, we'd have a similar equation, partial differential equation for the intermediate with diffusion, possibly decay, production, and catalysis. Okay, so so the idea is, why is this sort of self-organizing to the right size? You could imagine that inside a dense cluster, you want to have that form when the substrate is present. The one tricky thing here, and we'll come back to that briefly, is that we're imagining for the moment that this switching is allosterically regulated as an equilibrium process. Even though catalysis, of course, can be non-equilibrium, we want the switching to be equilibrium, so you're not spending energy to make this happen. And that means that we have to respect detailed balance. That is, for example, if an enzyme is in the middle of a cluster and it's got nothing allosterically bound, you might say, well, I want that to become switched to non-sticky, but it is now surrounded by other sticky enzymes. And so we have to point out that that makes it favorable for it to be in a sticky state. If you're surrounded by sticky friends, that might tend to keep you in a sticky state. And so we have to take that into account in the equations. It turns out it's quite possible to find parameter regimes where this works with allosteric regulation. I just wanted to emphasize that we're not cheating. We're not assuming mysterious fictitious energy sources for this switching. Okay, so let's look at the simplest case first, where I have some localized sources, and I want to know if my enzymes can cluster around those to be the right size. So I put a couple of sources in a box, and lo and behold, as I turn those sources on or turn them down, the enzymes cluster. I'll show you again. Enzymes are dispersed. You don't see them. We're only showing the high density regions. I turn on two sources. The enzymes find those sources. They cluster. They're different sizes because the sources are different strengths. And then I turn the sources down, and the enzymes spontaneously, some of them leave, spontaneously adjusting to give you the right size clusters. And the idea is that the size is set by the fact that these are just big enough to basically process all the substrate that's being released by those sources. And so you could say, okay, that looks nice, but how close to optimal is that? This is what we see. This would be the optimal distribution of the clusters. They look similar, but we can do better than that. We can actually calculate the efficiency of the pathway, how efficient the production of product is compared to the overall production of the substrate. And we can quantify that by how much better this is if you're clustered than if you're uniform. And what you can see here is that for this self-organized state, which is these gray diamonds in the main panel, as you increase the substrate decay rate over the enzyme catalytic rate, as you the substrates become more and more unstable, so that clustering around them to grab them all quickly is important, the efficiency goes up, and the efficiency of this self-organized state is pretty close to optimal, which is great. You only see there, well, maybe it's only up by efficiency is increased by a factor of 6 or so, but you can make that higher by, well, I didn't show that here. This has to do with the overall enzyme volume fraction. For numerical reasons, we work with a fairly high or quite high enzyme volume fraction of 10%. You see that at the top there. this efficiency basically scales inversely with that number. So if you have a 1% enzyme volume fraction, then the efficiency ratio goes up by basically a factor of 10. So and 1% or 0.1% are actually more realistic in real cells. Okay, so that was kind of easy though, because the substrate was being produced locally, and so all the enzymes had to do was cluster around it. What if the substrate is being produced uniformly? Now we want to form clusters where substrate comes into the cluster, turns into an intermediate, gets processed by the second enzyme in the pathway. Can we get that to work? And so here's a kind of quasi 3D version of that, where we have a uniform source within this tube, and the enzymes indeed are forming these clusters of these in 3D, doing this two-step pathway. I'll play it again. You turn on the source uniformly, and the system is organizing into sort of an optimally sized clusters. They're not too big, because then the substrate would come in, nothing would get to the middle, and that would be a waste. But they're not too small, because if they were too small, then the intermediate would escape before being processed by the second enzyme. So yes, it seems to be able to work. This is a lot of detail about that, basically showing you the same thing. The color plot there, though, is useful, showing you that you can get very, very high increases of efficiency, 100-fold, more than 100-fold in this scheme, assuming for things like unstable intermediates. Okay, all this requires energy. Where does it come from? And in this case, I've argued, well, the allosteric switching is not energy burning. It's respecting detailed balance. Where does the energy come from? Well, it actually comes from catalysis. So it's all being driven by energy released during this catalytic process. Not that there's anything added to the model, but that's where in the model the energy source is. And so you have energy due to switching. You're switching between these two states, and depending on the local chemical potentials, that could release energy. And then you have diffusion. And that's kind of also interesting, that this catalytic process, by turning this initial substrate into intermediate, is changing the concentrations of the substrate and intermediate in a way that also releases energy and leads to energy loss by diffusion. So you have these fluxes of sticky and non-sticky enzymes opposing each other. I'm going to do it this way, so that the total concentration at steady state of enzyme is fixed, but you always have a fluxes going down their preferred chemical potential gradients. I guess I should do it this way. Sticky guys moving in one way, non-sticky guys moving in the other way, dissipating energy. And so, of course, all of this does depend on the catalytic process being energy releasing. Okay, there are many open questions. What if less energy is available for catalysis? How to make these allosterically sticky enzymes? Love to chat about that. I think that's certainly possible, and I have some other ideas about how to do that. Love to talk to people. And what about, could we, if we don't like this idea of relying on an allosteric process, could we use an active process for switching? And that's not so crazy, actually, if you think about what happens with receptors on surfaces. They bind to some substrate or ligand, and that causes them to cluster. That's not very different from what we're proposing here. Something binds to an allosteric site and causes the enzymes to cluster. So we're not completely off target here. And I should say, there are many groups thinking about how to make clusters form. There's things called enzyme chemotaxis, which are certainly interesting. They're not exactly the same thing, or enzyme substrate affinity condensation. So there's a lot of interest in this topic in the field. Okay, summary. Channeling of metabolic intermediates by enzyme clustering can increase pathway efficiencies, reduce toxicity, et cetera. Lots of good things. We've argued that you could imagine activity-dependent phase separation or substrate-dependent phase separation can achieve nearly optimal pathway efficiencies. And the biophysical limits essentially set by detailed balance can be designed around or overcome by active switching. And of course, I want to thank people who actually did the work. Part one was Martin Jonica's lab and many postdocs and students, and part two, some overlap there of the theorists working on this idea about self-organized enzyme clustering. And of course, we're very happy to have gotten funded to do this work. And so that's what I had to say. I'm happy now to chat, answer questions, whatever we'd like to do.
Thank you so much. For the sake of time, I was not stopping you, but there are several questions in chat.
Sure. Maybe I should stop sharing at this point.
If you could read a question out loud before answering it, people
Sure.
watching the recording don't see the chat.
Yes. So I think I've answered some of these. Okay,
I
good.
think you're up to Tanner now. Tanner
Helgens. For your model one-step pathway, could you imagine it with RNA instead of peptides? The sticky-unsticky mechanism could probably be better to engineer if you have folding of aptamers instead of a peptide. Absolutely. Yeah, I mean, I'm very open to the idea of doing all of this with RNA. And since RNA can be both enzymes and of course RNA can stick to other RNA, that's great. Yeah, I'd love to chat about that. That would be fun. Great question. Next, Henrik Lewowski. Could you use the clusters to control reactions in two ways somehow? When cluster is formed, it's synthesis, and when the enzyme is unclustered, it would switch to hydrolysis, controlling switch between catabolism and anabolism. Wow, that is not something I've thought about. You could certainly imagine that this allosteric regulation of the enzyme could do more than regulate whether it was sticky. It could, in principle, also regulate what kind of catalytic activity it was carrying out. I'm sure that there are examples, I think I know of examples where different enzymes are allosterically regulated to change their activity, I think in nitrogen regulation. I'd have to think about what that would do for you, but it's a cool idea. So thank you. Pavana Asham says, awesome talk. Thank you. Love positive feedback. Could you use a membrane of a liposome for clustering instead of relying on self-clustering of the molecules? Well, you could certainly imagine, having a binding site for these. Yeah, I think I understand what you're saying. So instead of having the enzymes phase separate, could they, for example, stick to the membrane of a liposome when they're in the presence of their substrate? And I think the answer is yes, but again, I want to make sure that they localize to the right place, right? I want to make sure they, if you had these liposomes all over the place and only had substrate in some locations, then you could imagine the enzymes getting together on the right liposomes where the substrate was present. So I think that could work. I don't know that much about, how one would get things to stick to liposomes or the liposomes to be dispersed so that the clusters could form wherever they were needed, but in principle. Okay, moving on. Hagen Bailey, what self-organized do you mean? Why only the small size droplets fuse with each other instead of the big one fuse with small one? How does the enzyme to control the fuse process? Okay, so I mean that self-organized in the following sense, that these enzymes have two, in my model, have two states, one of which is sticky and one of which is not. The sticky ones are always going to stick to each other. And in principle, if you had all sticky ones, they would keep coarsening. They would not stop. The idea, though, is as the clusters get too big, for example, if I have a source of substrate somewhere and the cluster got too big, only the enzymes near the source would have substrate. So those would only be the sticky ones. The ones on the outside would not have substrate. They would not be sticky. So there's nothing keeping them there. They're just like any other protein in the cell. They would just diffuse away. So the self-organization is, if you're in a good place where there's substrate, you become sticky, and then you just keep growing until you've accounted for all the substrate. I hope that I made that more clear. Now, Aishwarya Juneja, sorry if I haven't pronounced that very well. Also positive feedback, thank you. I had a more general question. Does your model take into account the clusters formed by enzymes only? I assume not every enzyme molecule will necessarily bond to a substrate molecule. Okay, so that's part of the question. Yes, if the enzymes are not allosteric, there's nothing to bind to the allosteric site in our picture, they prefer to switch to a non-sticky state, and then they'll just diffuse around and do, not doing anything unless, now a new substrate comes along, binds the allosteric state, they become sticky, at which point they will stick to existing clusters or form nucleate new clusters. Another question in the same person. Also, EPIC1 as an IDP should phase separate and form condensates independently of Rubisco. Is that known? No, I would push back. Not all IDPs automatically form condensates. Nature has evolved certain IDPs to be sticky to themselves. EPIC1 is not particularly sticky to itself, but it has these very well-defined regions that stick to Rubisco. And you can see that in the sequence. Each EPIC1 has 5 nearly identical repeats, and those bind, specifically to the Rubisco. I understand, of course, there's a whole other literature of the IDPs that stick to themselves, but the EPIC1 is not one of them. I think I've come to the end of the chat.
Yes, I think you answered all the questions. I loved your talk. I was going to ask a question about in vitro reconstitution, but someone had beaten me to it.
Okay. Yeah, well, I'm happy to carry on later discussions with anyone here or who listens to the recording. I do answer my emails, so that's a good way to reach me. And I am particularly interested if people are interested in designing this kind of enzyme, whether it's a protein or an RNA. I'd love to see this actually happen, and I'd be delighted to collaborate with people on that. So, Kate, thank you so much for the invitation, and thank you all for listening. It's been a pleasure.
Thank you for a fantastic talk, and have a great rest of your Monday, and have a good week, everyone.
Very
Bye.
good. Bye, everyone.